So here, a close-up of a taste bud on the tongue. And there's many different kinds of taste buds, which we'll be talking, well, a few of them. And anyway, so the taste cells have these little taste hairs that allow the taste cell to be um, detecting what's in the mouth and to um, create this generator potential if it is the molecule that can activate this taste receptor. And of course, there's a sensory nerve fiber that can then transmit this information to the central nervous system. So we have 5 categories of taste buds, and one is salty. So for salty, we have salty is of course detecting like NaCl, right? So the way this works is that sodium can basically enter an ion channel. As it enters, it can then cause a depolarization. Depolarization, as we know, can lead to the opening of a voltage-gated calcium channel. So sodium is going to rush in with its concentration gradient. Calcium is going to rush in with its concentration gradient. When calcium rushes in, it'll bind to the it'll complex with some proteins that are involved and required for exocytosis, right? So calcium is required for exocytosis by binding to a protein and activating it to help with the exocytosis of neurotransmitter, right? And then the sensory nerve is going to be stimulated. So sour detects protons or hydrogen ions, right? And this can enter through an ion channel. And then what it's going to do is it's going to depolarize, right? Because these are both positively charged ions. It'll depolarize the taste bud. It'll lead to the opening of the voltage-gated calcium channels, and then the calcium will bind that protein that helps the neurotransmitter be released via exocytosis. Sensory neuron stimulated. In the cases of sweet and umami, which detect sugar or amino acids, umami for detecting amino acids, sweet for detecting sugars, they are going to enter the cell using a channel with facilitated diffusion. Then they're going to, as they bind their receptor, they're going to bind this receptor. There's going to be a G protein that gets activated, right? The G protein is usually binding GDP with the alpha, beta, gamma subunits. When the alpha subunit binds GTP, it'll then dissociate from the beta and gamma subunits, activate a secondary messenger. Sometimes it's cyclic AMP or cyclic GMP, right? There's other secondary messengers that get activated. You don't need to know what they are in this case. What the secondary messenger is going to do is it's going to close potassium channels. As they close, this will lead to depolarization. And then the depolarization will lead to the neurotransmitters being secreted via exocytosis and lead to an action potential. And then the last one to show you is bitter. So for example, quinine, which is found in tonic, is bitter. There's a receptor. When it binds the receptor, it can also activate the G protein. Okay, and the G protein will then do the same thing, right? It'll have exchange GDP for GTP for the alpha subunit. The beta and gamma will separate from the alpha subunit. A secondary messenger will be produced, and this will trigger calcium being secreted from the endoplasmic reticulum in the bitter cells, and that'll then help the exocytosis of neurotransmitter. And I believe here, I just want to add in that I believe calcium also plays a role in here, although it's not shown. Calcium is playing a role by complexing with the protein we've talked about before that helps exocytosis occur in the sweet and umami taste buds. So the olfactory receptors are located in the olfactory epithelium of the nasal cavity, and obviously it can detect volatile odors, and a lot of our olfactory receptors get damaged, and by just the environment, sometimes smoke or other chemicals we breathe can damage them. And there are basal stem cells that can produce new olfactory receptors in our nose. So if we look at the nasal passage, we see the olfactory bulb up here. And if we look at it close up, we see there are cilia on the different receptors for olfaction. And there's many, these have hundreds of types. In comparison to taste, we have the 5 types. There's like 340 types of different olfactory receptors. And if they detect their chemical, right, they can then basically depolarize and send neurotransmitters to these glomeruli, where they can secrete the neurotransmitters to these interneurons that will then be sending information to the central nervous system. So so the olfactory receptors have ciliated dendrites projecting into the nasal cavity, right? And these cilia bind to the odors. There's about, I said 340, but 340, 380, I've seen both numbers for different olfactory receptors. And it turns out that many odorants can stimulate the same receptor. And here's just a EM of one of the cilia and an olfactory receptor. So how do these work? So if the odorant binds the odorant receptor, right, we have G proteins again. What will happen is that the alpha subunit will exchange GDP for GTP. When it does, it'll separate from the beta and gamma subunit. In this case, the alpha subunit binding GTP will go to adenylate cyclase, which has the ability to create cyclic AMP from ATP. And then the cyclic AMP can go and bind a sodium-calcium channel and open it. So normally the sodium channel is sodium-calcium channel is closed, but cyclic AMP has the ability to open it, and the influx of calcium and sodium to the inside of the cell can lead to a depolarization event, right? So the odorant where there's up to 340, 380 of these specific odor receptors in our different types in our nose, and basically when the odorant is detected, it'll use these G proteins to activate adenylate cyclase to produce cyclic AMP that can then open the calcium and sodium channels, which will let calcium and sodium diffuse into the cell following their concentration gradient, leading to a depolarization and the secretion of neurotransmitters. So the next topic is vision. So vision comes from light energy, right? And we can only see a limited part of the electromagnetic spectrum. So we can see between 400 to 700 nanometers, right? From the blue to the red wavelengths. We cannot see ultraviolet, we cannot see infrared with our eyes. So if we look at the anatomy of the eyeball, I just want to point out a few key places. So this is the lens, right? The lens is connected to the other parts of the eye by the ciliary body that basically holds it onto the sclera, which is the connective tissue surrounding the eye that helps to give it structure and strength. In front of the lens, right, we have this anterior chamber, and in front of the anterior chamber, this is a fluid-filled chamber, we have a cornea, and then the iris of the eye is surrounding the center of the pupil. The pupil is, of course, where the light is coming through, and the iris can open and close depending on how much light there is in the environment to let just the right amount of light go into the eye. And inside we have the vitreous chamber, also full of this is full of vitreous fluid. And then in the back we have the retina, and this is where we have rods and cones that allow us to see. And here is the optic nerve, where the eye can send information, basically neurological information from action potentials down to the central nervous system. And basically the retina, actually I should say, is technically considered part of the central nervous system. The myelinated nerves here are myelinated by oligodendrocytes of the central nervous system and not Schwann cells. So when we see, it turns out our vision is so what you see on the right side of our body, that is projected onto the left side of our eyes. What we see on the left side of our visual field is going to be refracted onto the right side of our eyes. So our eyes are seeing things in inverted, and also it works not just left and right, it also works top and bottom. So what's on the bottom of our visual field is on the top of our eye, and what's on the top of our visual field is going to be refracted onto the bottom of our eye. So the brain needs to detangle all this information and have us view the world in the right conformation, where right is on right, left is on left, and up is up, and down is down. So the brain processing is extremely important for us to process vision and all the senses, right? Because all these senses are being transmitted from action potentials, right? But the brain knows where that action potential is coming from and the trigger for that particular neuron, sensory neuron. Pretty amazing stuff. So what I want to show you is how the lens changes shape for focusing on objects that are far or near. So for distant vision, the lens is going to be pulled taut. So basically the ligaments that are attached to the lens will be pulled by the ciliary muscle, okay? And it'll make it nice and taut, and this is very good for distant vision. However, if you're reading up close, okay, then basically we would want the ciliary muscle to relax. As you see here, it's relaxed compared to being contracted up here. And then we want the eyeball to have the lens to have a more round conformation. So you can see it's more spherical or flat for distance sight, but for very close, like reading something close, the lens should be more round. And one of the reasons that people over 45 have trouble seeing close is that the lens loses its flexibility. So when your lens is being pulled taut, when your ciliary muscles relax, it doesn't really do a good job of regaining the more round conformation for the lens.